a new start.
Great. Okay. So the recording has started now. I just want to. Well, I'm hoping it has. We'll see. Pause. Yeah. No, that's fine.
It seems it's recorded. So maybe just a word. So thank you, Professor Rachel Codd, for being here online for this lecture to the bioinorganic chemistry lecture at ENS. So we are in November 2024, and we are very happy to welcome you.
Thank you, Professor Polakar. It's lovely to be able to talk with students internationally. So very, very happy to deliver this presentation. So a little bit of my background. I'm from the University of Sydney in Australia, and my area of expertise, I've been trained in inorganic chemistry and bioinorganic chemistry, and my group is interested in using the tenets of bioinorganic chemistry to develop new technologies and to extend into new approaches towards discovering drugs and new therapeutic compounds. And so that's what I'm going to be talking about today, probably not so much on the radiotheranostics and diagnostics. All right, no, that's not progress. Oh, yes. So that's where I work. So I'm in an office that's sitting down in this corner here. This is the University of Sydney. It's a beautiful campus, and this is a jacaranda tree in the quadrangle. So if you're ever in Australia and Sydney, you can drop in and say hi. All right, so I'm going to start with this is a slide just to give you some context. And I'm going to begin with a clinical agent that was discovered in bacteria called a siderophore. So there is a lecture on the ESPIC site I checked from Professor Alison Butler, who's an international expert in siderophores, and I also am active in this area. So I'm going to begin just with a general discussion about siderophores. So these are low molecular weight organic compounds that are produced by bacteria, and the purpose is to acquire ferric iron. So almost all organisms, certainly humans and most bacteria and fungi, have a fundamental requirement for iron. And this is because iron is incorporated into metal prosthetic groups and cofactors, which form part of metalloproteins, which are survival critical, both for electron transport, respiration, and other key physiological processes. So for humans, we're able to acquire our iron from dietary sources, but bacteria and fungi aren't able to acquire iron in that manner, so they use a different system that they've evolved. So in response to low iron, which they sense using different biochemical cascades, bacteria will turn on their biosynthetic gene clusters to biosynthesize a siderophore. And what I've shown in this stick diagram here is an archetypal siderophore known as desferrioxamine B. So the bacterium will produce this compound and send it, which is really a ligand because it contains donor atoms that have an exquisite affinity to form a complex with, in this case, iron 3. So what I've shown in this very simple cartoon is the process for iron acquisition. So the yellow triangle represents the siderophore with no bound iron. This compound coordinates iron with very, very high affinity. And the iron siderophore complex is recognized by cell surface receptors that are upregulated at by the producing bacterium. And so this receptor recognizes this complex in the iron-loaded form. The recognition event then initiates a protein-mediated importation cascade where these compounds are actively imported into the periplasm of a gram-negative or into the interior of a gram-positive. And then ultimately the iron is released by mechanisms which can include hydrolysis and enzyme-mediated hydrolysis of the complex, or by a reductive mechanism, where a reductive environment or a reductase can reduce the iron 3 to iron 2. And under those circumstances, the affinity constant with the siderophore is drastically reduced and the compound falls apart. So in terms of relevance to, well, certainly medicinal chemistry, or bioinorganic medicinal chemistry, if you like, this compound called desferrioxamine B is used in the clinic. It's known as desferril, and it's used to treat a condition known as secondary iron overload. And this condition occurs in patients who are born with a genetic blood disorder called beta thalassemia. And these patients produce a hemoglobin that is functionally faulty. So there's a problem with the beta subunit of hemoglobin. And what that manifests as in terms of disease state is they suffer from anemia. Now, that may seem, you know, we often can use anemia, the word anemia, in quite a throwaway fashion, but in fact, anemia is really serious. And if you're not treated for anemia, you would eventually die. So what these patients have to do from about, once they're diagnosed, from about usually about the age of 6 months to 12 months, is undergo blood transfusions. So they receive units of red blood cells every 3 to 4 weeks. So that addresses the issue of hemoglobin, faulty hemoglobin. So now they have sufficient hemoglobin, but the issue there is that there is increased catabolism of these red blood cells, and that releases free iron in the body, and that can be toxic. So as secondary therapy, patients are administered what are known as iron chelators, which are these organic molecules that will hunt for the excess iron and then form an excretable compound. So these compounds have clear clinical significance in treating secondary iron overload. So in terms of how this works, what I've shown here is a 3D molecular structure of this compound, desferrioxamine B, in the absence of binding iron. So this compound has been evolved by nature to form an exquisite, very stable complex with iron 3. So and this complex forms by coordination with these 3 hydroxamic acid units. So this is a particular functional group known as a hydroxamic acid. And so what we have here is an N-hydroxylated motif bound together with a carbonyl group. So these oxygen donor atoms, and there are 6 in this coordination sphere, form a stable octahedral coordination complex with iron. And I've shown that here in the compound in the state where it's bound iron. So in the center here, we have the iron, and surrounding the iron, we have these 6 oxygen donors. So this meets the hard, soft, acid-base principle that we discuss frequently in bioinorganic chemistry to form a very stable compound. So with that backdrop, I want to touch on, that brings me to the lecture plan. So what I want to do is use bioinorganic chemistry knowledge to describe the development of a siderophore purification protocol. Then I'm going to look at this particular method and describe how we can apply this, use this to capture other metal binding natural products. We can use the method to produce natural product chemical probes, and I'll close out by discussing the method for the utility in drug discovery. So I thought this was a good topic because the method that I'm about to describe is easily understood, and it is underpinned by the tenets of bioinorganic, or certainly coordination chemistry. And then really part of the purpose is to try and invite the notion of both creative thinking and lateral thinking. So if we develop a method that in itself is novel and useful, then we might start asking the question, what else can we use this method for in terms of driving discovery and driving new approaches to discover new drugs and new drug targets. All right, so just to refresh, we're going to develop a siderophore purification protocol. Now, the need for this arises from the fact that siderophores have quite complex structures and they are difficult to make in the laboratory using standard organic synthetic chemistry. For some siderophores, this has been achieved, but these syntheses are usually multi-step and generally quite low yielding. So if we're interested in studying siderophores, which are relevant in terms of understanding how bacteria acquire iron and therefore are relevant in terms of potentially being able to develop antibiotics, then we need to access these molecules for further study. Now, because they are produced in bacterial culture in very, very low yields, we need to design a clever purification protocol. So that brings me to the method that really underpins what I'm going to be talking about in this lecture, and this is immobilized metal affinity chromatography. So those of you who have a biochemistry background or a molecular biology background, and others may also have heard of this method, I'm going to describe the method as it's used standardly in its standard format. So this is a method that's used globally in laboratories for recombinant protein purification. And I'm sure some of you, some students and perhaps Professor Polakar have used this method. So what we have here is we have a solid resin, and attached to that resin is a covalently added tridentate chelate. So this tridentate chelate is aminodiacetic acid. So that's shown in this part here. So aminodiacetic acid itself is bound to a nickel ion. And so the resin format is partially saturated. So we have 1 unit of nickel per 1 unit of aminodiacetic acid. In the other parts of the coordination sphere, we'll have will be occupied by, as I've shown here, water molecules. So the way this is used, and if you've used immobilized metal affinity chromatography, which is shortened to IMAC, or you'll know that it's green, and it's green by virtue of this coordination complex that's immobilized at the resin interface. So what we do is, in standard use, we'll produce recombinant protein and we introduce a histidine tag either at the C or N terminus, usually at the C terminus. Now, histidine, the imidazole groups of a histidine amino acid residue have a high affinity towards nickel, and the histidine residues are configured in a manner where they displace the water molecules from that nickel aminodiacetic acid coordination sphere and then create this hexadentate saturated coordination sphere. So essentially this histidine protein is now adsorbed using affinity chromatography to the nickel center. All of the other material can be washed through the resin. So, and then we're positioned to displace our target protein. And so we use a competition process where we introduce high concentrations of imidazole in the buffer, and that will outcompete the bound histidine tagged protein, and that will displace our purified protein and liberate an imidazole saturated aminodiacetic acid compound in the resin. So this is really, really well understood. And when I was thinking about an approach to purify a siderophore, I wondered whether we could actually apply this method to a siderophore. Because what we understand about siderophores is siderophores bind metal ions. So siderophores have been evolved in nature to bind ferric iron, but the hydroxamic acid unit in a siderophore also has affinity to other metal ions, including nickel. So really the question we were asking was, might it be the case that we could, rather than absorbing a histidine tag protein, could we actually take a siderophore, and we posited that what would happen would be that this siderophore would displace the aqua ligands in this nickel aminodiacetic acid complex and be retained on the solid phase resin. We would wash off all the unwanted components, and then we could introduce imidazole and displace our purified siderophore. So this was the concept that we wanted to explore, remembering that the utility was that we were going to potentially be able to harvest very, very low levels of siderophore from a bacterial culture. So what I've shown here are the results of us developing this method. So on the left, I've shown a culture of a bacterium known as Streptomyces pilosus, and this is the bacterium that produces desferrioxamine B as its native siderophore. And this particular bacterial strain, or a super producer, is used by companies, industrial companies that ferment large-scale production of this particular bacterium to produce desferrioxamine B for clinical use. So we acquired some of this bacterium and cultured it, and showed in other experiments that it was producing DFOB. We centrifuged down the system to separate the supernatant from the cell pellet, and we ran a liquid chromatography experiment. So in the upper trace in A, this shows the entire, well, certainly the soluble metabolome. So all of the metabolites that are present in that bacterial supernatant. So this is going to include natural products that have been biosynthesized by the bacterium, in addition to salts and other components in the culture medium. We took that raw supernatant and all we did was adjust the pH to pH 9. So what we're doing by adjusting that pH to a more alkaline value is deprotonating the hydroxamic acid units, which have a pKa value sitting at around pKa 8.5 to 9. And then we wash the resin to remove any unwanted components, and then we displaced the bound components by competition, and then reran the liquid chromatogram. So what you can see moving from panel A to panel B is an extraordinary purification of this crude mixture. So it's difficult to accurately predict how many compounds are in panel A, but conservatively, I would say at least 100. So with a very, very simple procedure, which is based on the affinity of a siderophore for the hemisaturated nickel aminodiacetic acid complex, we have selected out desferrioxamine B and an analogue called desferrioxamine A1. So in order to prove that these are siderophores, what we can do is take this mixture and introduce some iron 3 plus, into that mixture, and then reacquire the chromatogram. So if the posit is that these are siderophores, then we would expect these compounds to form complexes with ferric iron, and we would expect those ferric siderophore complexes to have a different retention time on the liquid chromatogram relative to the free organic compounds themselves. So that's what's shown in moving from panel B to panel C. So these signals here I've indicated in an orange, which correspond to the desferrioxamine B in its iron-loaded form and desferrioxamine A1 in its iron-loaded form. So what I've done in this lecture is just popped in a couple of questions and discussion points, just in case, you know, we might want to discuss this a little bit further. And feel free to put your hand up or put some questions in the chat. So I thought that we could talk about what you might conclude from the traces in B and C, because what I've really told you is that we took this mixture and we ran it down this purification system, and indeed we did select for our target compounds. But you can also see that we selected for, there are 2 other signals in panel B, and when we added iron, the signals at that's eluting at about retention time 9 minutes actually became resolved into 2 signals. So what that's telling you is that there are actually more likely to be two species, at least 2 species, that are almost co-eluting in this signal at 9 minutes, and that when we add iron, these signals are resolved into different species by virtue of coordinating iron. So it's really the purpose here is just to try and get some rigor about interpreting a result such as this. So there's a number of things that you can conclude in terms of the number of species and also whether or not they have an affinity for iron. So the species that's eluting at 14 minutes doesn't seem to have shifted in the presence of iron. So you might reasonably conclude that this species has no affinity towards iron. Whereas the 2 species in the system where we have not yet added iron become resolved into 2 signals upon the addition of iron. And so that's telling you that whatever 2 species are hidden under this curve do have an affinity for iron. And what I can tell you is that these species are amino acids, which are present in very high concentration in the bacterial culture medium. So from memory, I'm not going to quite get it right, but I know that there's a phenylalanine and a tyrosine, and I forget what the other amino acid is, but certainly it's described in some of our work. It's not so important. It's really just an approach to interpreting these results. All right. So if we reflect on why this could potentially be useful, this method, it enables us to culture any bacterium and undertake an analysis of its entire siderophore metabolome. So most bacteria will produce one type of siderophore, and it's generally the case that bacteria produce a chemically unique siderophore. And the reason for that is because if a bacterium produces a chemically unique siderophore, then it follows that the complex between that siderophore and iron is also unique, and it will have a unique fold and a topology and 3-dimensional structure. And that will enable recognition by the receptor at the cell surface, which will have an amino acid structure that has evolved to recognize just that single iron siderophore complex. So the reason for that is for evolutionary competition. So bacteria are in competition with each other for very limited iron. And so from an evolutionary point of view, a bacterium will want to produce a siderophore that is unique to its own species. And so another competing bacterium cannot access the iron that the parent bacterium has expended significant biosynthetic energy in producing the siderophore and also the receptor. It is not universal that a bacterium will produce a single siderophore. So in some cases, bacteria produce multiple siderophores. And this is certainly the case in this study here, where we took the IMAC method, so the immobilized metal ion affinity chromatography method, and used it to interrogate the siderophore metabolism of a marine bacterium called Salinospora tropica. So Salinospora tropica is a topical bacterium at the frontier of natural products discovery. And the reason is because its genome, almost 10% of its genome encodes for natural products that have been declared novel or unknown using genome mapping. So we were interested in what sort of siderophores this bacterium might produce. So we obtained some of the bacterium and cultured it, and it's got a really interesting cell morphology. And then we took the supernatant and used IMAC to purify out the siderophores and ran an LC trace. And what you can see here is that this particular bacterium does produce a large range of siderophores, which was quite surprising to us. So it does produce desferrioxamine B, which is shown down in this region of the chromatographic trace. But really what's relevant here is that it produces all sorts of different analogues of this siderophore class. So you might ask why. What would be the rationale for producing so many different siderophores that essentially, in a global sense, have a common function in terms of ion acquisition? So what we did with this bacterium is we undertook culturing under different conditions, so under different temperature and different pH values. And this is really representing the marine environment, which is quite has diverse microenvironments. So this bacterium is resident in tropical waters, which are quite warm, and the pH value of oceanic water can be varied. So what we were querying was whether the rationale for this bacterium in producing many, many siderophores, could it be that some siderophores were better tailored for particular environmental conditions? And what we found by mapping the siderophore metabolome under different culture conditions was that the relative concentration of these siderophores changed as a function of environment. So that indicated that it might be the case that a bacterium, when it finds itself in a particular environmental niche, will preferentially produce a siderophore that, for example, has exquisite affinity for iron under more alkaline conditions, or if it's at lower temperature, it might produce a different type of siderophore. So I think of this as akin to having different sorts of cutlery depending on what sort of meal that's put in front of you. So you might have a knife and fork for a steak, or you might have chopsticks for noodles, and so on. So you're still accessing your nutritional requirements, but you choose what sort of receptacle or tools to use to actually acquire that. All right, how are we travelling? All okay so far? Yeah. All right, I'll just plough on. So going back to IMAC and revisiting the notion that this is built on inorganic chemistry. So what we know about inorganic chemistry is that while the base resin contains nickel, so if you were to buy IMAC resin, the most common commercial form is nickel. What we also know is that copper can also bind to this ligand type, which is known as polyamino carboxylic acids. So we could potentially purchase a different type of resin, which has this tetradentate ligand, which is known as nitrile triacetic acid. And rather than using nickel, we could absorb or load copper into this system. Why would that be useful? We thought that we could use copper to capture different sorts of bacterial metabolites that we know have an affinity for copper. Alternatively, we could use cobalt as the metal ion to capture different types of ligands, or we could use zinc. So essentially what we're asking here is we're switching both the metal and the type of polyamino carboxylic acid and screening this against different bacterial cultures to capture compounds that may be of interest. All right, so this is another question that I wanted to pose. So if we just touch for a moment on charge, if we look at this resin I've shown here in panel A, we have a nickel in the +2 oxidation state, and we have an amino diacetic acid resin, which has a 2 negative state because these carboxylic acid units are deprotonated. And so the overall charge of this resin is neutral. In the case of the copper resin, we have a copper, which is copper 2+, and then we have this tetradentate trianionic ligand, because we've got 3 deprotonated carboxylic acids that are bound into the copper. So the charge of this immobilized unit overall is uni-negative. And we can use that similar logic just to determine the overall charge of these different IMAC formats. The point here is to consider, when you're actually conducting an IMAC experiment, the principal mechanism of action is affinity chromatography. So we're banking on an exquisite binding constant between the target compound of interest and the metal ion, the immobilized metal ion. What we don't want is to complicate the binding procedure by any other types of attractions, so such as ionic interactions. So for that reason, we include high concentrations of salt in our buffers. And so that will abrogate any electrostatic binding interactions and only deliver true and correct results that are binding by virtue of affinity chromatography. So in terms of looking at a different metal ion, our compound of interest was an anti-cancer drug that again is clinical, and it's known as bleomycin. So bleomycin is produced by a bacterium called Streptomyces verticillus, and this is in use in combination therapy for treating testicular cancer. So this is one of 3 drugs that is used for testicular cancer, which has resulted in a major improvement in prognosis for these patients. So it's used together with cisplatin and an etoposide inhibitor, and it's really transformed patient outcomes. So in terms of its mechanism of action, when it's administered to the patient, this region of the molecule binds iron in the iron 2 oxidation state, a ferrous ion. This part of the molecule intercalates in DNA, and there's a redox mechanism that actually causes damage to the DNA. Interestingly, when Streptomyces verticillus produces this compound, so it turns on the biosynthetic genes and undertakes the biosynthesis of this compound, the purpose in terms of the bacterium itself is it produces it as an antibiotic. So if Streptomyces verticillus finds itself in competition with a different bacterium, it will upregulate the biosynthesis of bleomycin and send that out to the extracellular milieu, and that will act as an antibiotic against the competing bacterium. But when it actually produces it as the copper complex. And so understanding this, we thought that we could potentially use copper-based IMAC to purify bleomycin from cultures of Streptomyces verticillus, and that could potentially be useful in terms of improving or streamlining pharmaceutics production. So similar to the experiment that I showed you with the desferrioxamine B, this is really a quite extraordinary purification profile of bleomycin. So we obtained some Streptomyces verticillus and cultured it, and showed that it was in fact producing bleomycin, and using very, very similar notion to what I described previously, we took that supernatant prepared a copper-bound immobilized metal ion affinity chromatography matrix. We had to first remove any iron that was present in the bleomycin complex to make sure that it was metal-free, because if the copper was already loaded into this compound, it would not bind to the iMac resin. So we undertook that process of metal removal first of all, and then we bound it to the resin, the copper resin, and then we washed off all of the unwanted components and then eluted the bleomycin. So what's impressive about this particular piece of work is that what I've shown in this chromatographic trace marked A is the metabolome, the whole metabolome in terms of the supernatant. So you can see this is really, really complicated solution. Like in terms of actually isolating each one of these species using regular reverse phase liquid chromatography, it would be very, very difficult. What we did was shown in the panels on the right-hand side is really just the map of the whole iMac procedure. So here we have the whole supernatant, and these panels in B, C, and D and E are when we are binding them to the resin. And then we wash the resin exhaustively until we can see essentially the chromatogram is close to baseline. And then we elute the compounds using an elution buffer. And what you can see here is really this panel is the key take-home point, that we get extraordinary purification from this very complex mixture. And we were able to purchase some commercial bleomycin, and we undertook a standard addition experiment where we add some authentic verified bleomycin to the system, and we can show that these indeed are bleomycin. So what we've seen so far is using simple notions of inorganic chemistry and coordination chemistry, and to push those into utility for potential industrial production methods for current clinical agents that are critical for global health. So pushing this notion into another realm, if we return now to the first resin that we talked about, which was our nickel resin, which has desferrioxamine B bound to the resin via coordinate bonds. Now, what's noteworthy about desferrioxamine B is that I've described the fact that it has these 3 hydroxamic acid units as part of its backbone, but it also has a pendant amine group. And this amine group, so you can see here, I've drawn it here as a cationic species. This amine group is not involved in forming the complex with iron 3+. So it has a function in biology really to make sure that this compound is water soluble, because it will be charged at physiological pH. But the amine group is not involved in the function of binding iron. So what we considered would be interesting to test was using an iMac resin as a solid phase organic synthesis matrix. So those of you who have done sort of entry-level chemistry or some organic or medicinal chemistry would have or may have heard of solid phase organic synthesis. Certainly those of you who have been involved in producing peptides, who may have used an amino acid synthesizer or had classes on this, would recognise that when we do synthetic chemistry using a solid phase resin, that can be very helpful, because what we can do is we can drive reactions to completion by introducing reagents at high stoichiometric excess. So that's very useful. And further, we can wash impurities and those reagents that may be in excess just with wash steps. So the value of solid phase organic synthesis is based in efficiency of synthesis. So we can, rather than using more traditional solution phase synthesis, where you might produce intermediates and you need to purify, and that can be complicated, and then you undertake another synthesis in solution phase and so on. So in this particular piece, what we wanted to determine was whether in fact we could broaden the function of an IMAC resin and use it for solid phase organic synthesis. And that would be unusual because ordinarily solid phase organic synthesis uses resins that require a covalent bond between the first building block and any building block that's subsequently added. What we're proposing here is that instead of using a covalent bond in terms of the reagent, the primary reagent, which is in this case is desferrioxamine B, we're using a coordinate bond. And the point here is that because we have multiple coordinate bonds, we considered that from a functional point of view, multiple coordinate bonds, which are by definition weaker than a covalent bond, but we thought that cumulatively, multiple coordinate bonds could be as strong as a covalent bond. And if that was the case, it might be possible to undertake some chemistry at this amine terminus. So we undertook a simple study of seeing whether we could produce biotinylated desferrioxamine B. And that's shown in this structure down at the bottom of the slide. And well, I'll get to that in a moment. So what I've shown here is the proposed scheme. We were going to load nickel onto an IMAC resin, react that solid phase system with an activated biotin motif. And what you're looking for here is the formation of this amide bond. And then we could use very mild chemistry to release this target compound from the resin. So the question that I wanted to pose for this section is what might you use this type of chemical probe? So essentially what we've done here is created a chemical probe. Why might this be useful? And so what it could be used for is, you may recall from the opening slide, I showed a cartoon of siderophore-mediated ion uptake. And the siderophore is a key component of that ion uptake pathway, but of equal importance is the outer membrane transporter that is expressed at the cell surface of a gram-negative bacterium. And also there are proteins sitting in the periplasmic space that transport the ion siderophore complex, and there are other proteins involved in ultimately delivering ion to the cytoplasm, and also sort of functionally similar proteins in gram-positive bacterium. So what, in theory, what we could do is use this particular this chemical probe as a bait molecule in a pull-down format to select for proteins that recognize the ion DFOB motif. If we introduced ion into this biotin DFOB system and took this chemical probe and incubated that with a proteome of Streptomyces pilosis, for example, and then we got a streptavidin immobilized resin. So streptavidin is a protein that recognizes biotin with very high avidity. Then we could use that resin to actually capture whatever protein was bound to the ion DFOB system. So essentially we can use these biotin siderophore compounds as molecular bait to discover cognate binding proteins, and these cognate binding proteins are potential antibiotic targets. So these could have some utility. All right, so here is the result of this experiment. So we used two approaches. We used two methods of detection. So on the left-hand side, we're looking at solid-state electronic absorption spectra. And on the right-hand side, we're using a reagent 2,4,6-trinitrobenzene sulfonic acid, which forms an orange complex in the presence of a reactive amine group. And so we've taken a nickel-free resin. So this is a resin that simply contains covalently bound amino diacetic acid. And we would not expect to see any absorption in the visible region of the spectrum. There are no free amine groups in that matrix, and so we would expect to see a negative result in the presence of this colorimetric agent. The next step is we adsorbed nickel onto the amino diacetic acid resin. So we're now forming nickel IDA, or a standard IMAC resin. And what you can see here is if we take that resin and run a solid state electronic absorption spectrum, we see a signal at about sort of 620 nanometers, which is representative of that nickel IDA complex. The resin, when treated with the colorimetric agent, does not stain positive, which is compatible or consistent with the fact that there's no primary amine present, reactive amine present. What we did next was we loaded our desferrioxamine B to the nickel resin. So this is where we're forming our, we've taken a hemi or partial or half saturated nickel coordination sphere, and then we're introducing desferrioxamine B, which is then saturating that octahedral nickel coordination sphere with 3 hydroxamic acid units. And that's shown by this spectrum in panel C. Now, because we've bound DFOB and we are positing that the amine group remains reactive, meaning that we could potentially do some chemistry at that amine group. And that is confirmed by virtue of the fact that the resin turned bright orange when we reacted it with the colorimetric agent. And what I've shown here in the solution is just a reaction between solution desferrioxamine B itself and the same reagent. What we did in panels D and E, in panel D, we prepared some biotinylated desferrioxamine B using traditional solution phase and purified it. And then we adsorbed that onto a nickel-loaded resin. So this panel D is really our positive control. This is what we would expect if our coordinate bond-dependent solid phase organic synthesis system was successful. So panel D, we have a modulation in the electronic absorption spectrum. And by virtue of the fact that we've now masked that reactive amine with a biotin group, so we've now formed an amide bond, we would expect the resin to no longer be reactive with the colorimetric agent, which is shown here. And then panel E is the compound that we actually accessed using what we've called coordinate bond-dependent solid phase organic synthesis. So this was really the ultimate test of the experiment. And you can see that there's a good match between our positive standard and our experimentally produced compound. In terms of looking at the purity of the desferrioxamine or the biotinylated desferrioxamine B, what we've done here on the left-hand side is a reverse phase HPLC. On the right-hand side is electrospray ionization mass spectrometry. We're looking at positive ions. And we can see the solution phase we would expect, which has been purified, so a reasonably pure system that characterises as the sodiated adduct of biotin DFOB. In terms of the product that we produced using coordinate bond-dependent solid phase organic synthesis, this was very successful in producing a very clean signal that mapped to what is essentially a standard, and we obtained the same value for the sodiated adduct. In panel C, what we were trying to demonstrate was the value of and the role of the nickel in the resin. So we actually ran in parallel this exact same system, but using aminodiacetic acid resin that contained no nickel. And essentially, this was to prove that the operation of this method was occurring by virtue of the coordinate bonds between desferrioxamine B and the nickel on the resin. So this is if we don't have the nickel, then essentially what we're doing here is operating under solution phase conditions, and we see a more complex system of reagents that have not reacted, and in terms of the ESI mass spectrum, we see some unreacted NHS biotin, both as different adducts. So this is going to be the protonated and this is the diprotonated, some unreacted desferrioxamine B, and very low levels of our target compound. So in essence, this shows really what these data show is that the coordinate bond dependent solid phase organic synthesis has potential in producing chemical probes of natural bacterial metabolites that have an affinity to a metal ion. Just a bit conscious of wearing you all down. All right, so we're in the home straight. What I'm going to talk about next, we are going to take IMAX, so the notion that we've talked about in the lecture to date thus far, and now we're going to push a little bit into drug discovery. So I'm sure that in other classes that you, and certainly a big field of bioinorganic chemistry, are metalloenzymes. So this is a field that, almost all bioinorganic chemists find very interesting and very enriching. And what we find is metalloenzymes comprise about 40 to 50% of the human proteome. So to understand metalloproteins is really, really important in understanding human physiology, and certainly the physiology and mechanism of almost all life forms. And many of these metalloenzymes can become dysregulated in disease. And so by definition, that makes dysregulated metalloenzymes a drug target, meaning that if a human has a metalloenzyme that is operating in an aberrant manner, so say, for example, the enzyme known as HDAX, which is histone deacetylase, in cancer, histone deacetylases can be upregulated, and that's actually problematic in terms of cancer development. And so if we inhibit a histone deacetylase, then we could improve patient outcomes for particular types of cancer. So that's the way we think of drug targets. So histone deacetylases contain a zinc ion at the catalytic active site, and this zinc ion is critical for activity. A zinc ion is also present in matrix metalloproteinases, which are another cancer target, and it's also present in angiotensin converting enzyme 1 and angiotensin converting enzyme 2, which you may recognise as part of the mechanism of the COVID virus. Other metal ions that are present in different types of metalloproteins include copper-2, which is present in tyrosinase, and ferric ion, which is present in 5-lipoxygenase. So this is just to give you a sense of the presence of metal ions in metalloenzymes in humans and the different types of metal ions. And this particular article by Seth Cohen, who does a lot of work in metalloenzymes, and this is a really nice review paper if you've got time to read this. This paper describes that in 2017, 4% of the new chemical entities approved by the United States Food and Drug Administration were metalloenzyme inhibitors. So that's a really significant impact in terms of drug discovery. So that is the backdrop for me leading into the next closing stage of the lecture. So we proposed that given that the metal aminodiacetic acid scaffold is hemisaturated, meaning that the compound stoichiometry is one aminodiacetic acid unit to one metal ion, then that was reminiscent of a metalloprotein active site. Now, to prove this, what I did was looked at some drug complexes, or looked at the crystallographic database for drug ligand complexes, and mapped these onto crystal structures of nickel IDA and other constructs, and showed that there was excellent overlap. So that's probably at this point a little bit confusing. What I've done here is I just widened out the scope of this concept. So we've got zinc IDA, iron IDA, nickel IDA, and what I've done is I've trawled through various compounds that actually bind to these systems and shown that there is high coincidence in terms of atomic overlap. Really, I think that's probably very confusing. What I'm trying to suggest here is that in a metalloenzyme, the metal ion at the active site is coordinatively unsaturated. And if you ask yourself, why is that so? The answer is because that metal site needs to interact with the substrate. And so in many metalloenzymes, not all, but in many, the substrate will actually form coordinate bonds with that metal ion, undergo the transformation as mediated by that metal ion and the metalloenzyme overall, and then be displaced from that metal ion at the active site. So in this work, or in this study, we posited that the IMAC coordinatively unsaturated unit could be reminiscent or represent a similar state of a metalloprotein active site. And if that was the case, then we could potentially use IMAC as a surrogate of a metalloprotein active site and take mixtures of compounds, either known compounds or unknown compounds, and use an IMAC system to select for a given compound. And that particular compound that might be absorbed onto that resin could potentially be a new metalloprotein inhibitor. So I hope that's not too confusing. It might become clearer as we move through. So to test this, what we did was we took a mixture of 17 compounds. So this was really a proof of concept study. So what I've shown, and so here are our 17 compounds. And what we have, we selected a group of clinical compounds and non-clinical compounds. So we've got an angiotensin converting 1 inhibitor, another ACE1 inhibitor. We have HDAC inhibitors, some clinical, some experimental. We've got 5 lipoxygenase inhibitors, and tropolone, which is a tyrosinase inhibitor. and we included in this mixture a compound that's an antibiotic that doesn't necessarily have a known function in terms of binding metal ions for its activity, but nonetheless, it has a chemical motif that we would recognise as having capacity to bind to a metal ion. So it's really what you would describe. These compounds, 1 through 16, contain functional groups that are known to bind metal ions, and that is important to their function. Compound 17 doesn't have a known metal ion binding purpose in its function, but it does contain a motif that we would recognise as metal, having the capacity to bind a metal ion. So we mixed all these compounds together, and then what we did was we set up a panel of IMAC resins in parallel. So we prepared a copper resin, an iron resin, a gallium resin, a nickel resin, and a zinc resin. And then we adsorbed the mixture on each of these columns, washed off unbound components, and then eluted components that were selective. And then what I've shown here is just a heat map of the results. So in designing an experiment such as the one I've just described, we needed to create really create some measurement parameters. And I think this helps in terms of looking at the results to come. So if we imagine a resin that was entirely successful in binding a given compound, what we expect is if we adsorb a compound onto a resin, when we're binding, we don't expect to see any of that compound in the eluent because the compound is actually bound to the resin. When we elute the compound, so here we're actively trying to displace the compound from the resin, then we see the entirety of that compound. So there's 3 things to consider. First of all, if we adsorb 100 units of that compound onto the resin system, and we can count 100 units of that compound in the system overall once we've done the experiment, then we would describe that as having an accounting ratio of 1. So it's 100%, it's perfect. We haven't lost any of the compound, is essentially what that's saying. If we have this system in panel A, which is the most successful outcome, where all of the compound has been successfully retained on the resin and successfully eluted, then we would have a resolution ratio or a retention ratio of 1. That's perfect. And then we created something called the performance ratio, which was simply the multiplication of the accounting ratio and the retention ratio. So this is the ideal situation. If we reverse if we go to the other extreme, and this is an undesirable situation, then what happens is when we try binding the compound to the resin, none of it binds, right? So it all just falls through. And although the accounting ratio is 1 because we've accounted for all of the, 100 units of the compound, the retention ratio was 0. There was none that was bound upon elution. And so the performance ratio then becomes 0. We could have, and these other 2 situations show slightly different notions where we have some binding and we have some elution. So this is a partial affinity. So we can account for all of the compound, but not all of the compound has been retained. And so the performance ratio is reduced. And in this particular scenario, we've got some of the compound that's bound, but we can't account for all of the compound that we loaded on. So that's really telling you that there's some sort of phenomenon going on at the resin. For example, it might be precipitating, or for whatever reason, we can't seem to elute the compound. And so again, using this measurement system, we would declare this as a performance ratio of 0.1. All right, so I've really answered that question. So this is pretty messy, but we can try and talk this through. What I've shown in the panel A is panel A and panel B. These are really the standard curves. So this is really just a liquid chromatogram of the mixture itself, right? It hasn't actually been processed by a resin. And this particular system in panel B also hasn't been processed by resin. But the difference is that in panel A, we've mixed the compounds in the buffer system used for adsorption. And in panel B, we've mixed the compounds in the buffer system used for elution. So this gives us a good measure of how the compounds themselves are going to perform. Now, on the left-hand side, we've got all our different resin systems. So copper, iron, gallium, nickel, and zinc. And these, when we ran the solution through a copper resin, for example, you can see if we just focus on compounds 9 and 10, they are not appearing in the binding fraction. So that would suggest that they may be binding to the resin. There's some binding here, but not a great deal. So without going into too many details because it's too confusing, if we look at some really clear results and focus on compound 15, then in the case of compound 15 and the selectivity of binding, this bound in a close to selective manner to the iron resin. It didn't bind to the copper resin. It bound in very low levels to the gallium. It didn't bind to the nickel, and it didn't bind to the zinc, or at very low levels. And you can see this appearing in the unbound fraction. So this is trending towards the fact that compound 15 is selective for the iron IMAC resin. So in a discovery sense, if we used an iron IMAC resin, we could screen complex mixtures, and compounds that actually bind to that iron resin could be potential metalloprotein inhibitors of 5-lipoxygenase, for example, which has an iron at the catalytic active site. What I've shown here is compound 17, which is the antibiotic, which had an incidental metal binding group. And indeed, this antibiotic bound to the nickel and the zinc resin, and to a lesser extent to these other resins. And so that shows, potential capacity for these systems to be used in discovering compounds that have an inherent metal binding capacity. And then what you would really need to do is what's called a phenotypic screen to see, which is a broad screen to actually check for activity either as an antibiotic or as some sort of metalloprotein inhibitor. And what we did in this work was calculate all of the accounting ratios, retention ratios, and so on, and then ultimately came up with a selectivity ratio. And really, because this does get very confusing, if we just look at this, so this is a heat map, and in this box here, what this represents is that compound 15, which was our 5-lipoxygenase inhibitor, had an entire selectivity for the iron 3+ resin. And that was consistent with the fact that 5-lipoxygenase contains an iron at the active site. So what are some of the advantages of this approach for discovering metalloenzyme inhibitors? It's very simple. It's easy, it's water compatible, and it would be amenable to robotics and to high throughput an approach. And I'm conscious of time and wearing you down, and So just to skip through this, we are using this method to actually screen whole metabolomes. And we've gone back to Selenospora tropica, using a workflow where we use the IMAC resins and we can map out enrichment, match mass spec patterns to calculated patterns, and then identify compounds and go into genomes and actually de-replicate and discover novel compounds. And these data, which is unpublished, is just showing us using this and selecting for compounds that are enriched either on a copper resin or a nickel resin or a zinc resin, and we're in the process of finalising out this data. So what might be considered some disadvantages? So if you think about the geometry of a aminodiacetic acid motif, then that will deliver facial coordination. So it's not necessarily, well, it's not certain, but it will skew away from compounds that may bind in a meridional fashion. So say, for example, a natural product that has an aromatic motif in terms of binding, that may be less amenable to being selected on an IMAC resin by virtue of the conformational flex of that aminodiacetic acid chelate. Second, in all the discussion that I've described, we need to be conscious of what's called transmetallation. So if you actually bind a compound that has a very, very high affinity for a given metal ion that is immobilized as part of an aminodiacetic acid compound, or you introduce a compound at very high concentration, then you may observe that compound actually not being retained on the resin, but rather appearing in the eluent in the metal loaded form. So essentially it will strip the metal ion, or it has the potential to strip the metal ion from that resin. And that is not helpful in terms of retaining that compound. All right, so this is my lecture summary. What we've talked about today is the operation of immobilized metal affinity chromatography and the fact that this is founded on the principles of coordination chemistry. What we've undertaken is the use of these fundamental tenets, so understanding of coordination geometry, pH, pKa, charge, and how we can use this to develop new IMAC-based methods to produce chemical probes, to discover new drugs, and as inhibitors of metalloenzymes. And really, if you think about what this distills down to, it's looking at the merit and impact of cross-discipline learning. So this is taking a very traditional background in inorganic chemistry, pushing into microbiology, pushing into drug discovery, pushing into technology development, pharmaceutics production, generating chemical probes, and so on. And really, it's pushing to think creatively, which is what we really enjoy as academics and students and innovators of the future. And so I just wanted to close with just a comment on this is our beach house down on the south coast. And often we'll have students that come from overseas and sort of say, is it true that you have kangaroos walking down the main streets in Australia? And ordinarily I would say no, but at our beach house, the answer is yes. So this is our backyard, and this is actually an enormous kangaroo, and he's just relaxing, having a bit of a rest. This is our front yard. So in some ways it is true. And so I will leave it there. And okay, so I will stop recording.